So you will develop more vessels to feed the working muscles. Increase the myoglobin concentration in the muscles. Increase the mitochondria in the muscle cells. There will be an increase in all of the enzymes associated with cellular respiration. So the muscles are becoming more efficient. There'll be an increase in endurance. There will be very little increase in muscle mass, but there will be an increase in muscle tone. So aerobic exercise does not bulk you up, but everything gets firmer. All right, I'm going to move the chart over here to the left board, so just continue down here if you have room. And Madeline, you holler at me if I forget to go back to the big board. Will be a decrease in the triglycerides. These are the circulating triglycerides in your blood, and we don't want a lot of those. That's one thing that leads to heart disease. A decrease in total cholesterol. A decrease in LDL. LDL is the form of cholesterol that we consider the bad stuff, the stuff you don't want. There will be a slight increase in the LDL. I'm sorry, the HDL. That's the form of cholesterol that we consider the good stuff, the stuff we do want. Your resting heart rate will go down. And that's probably the easiest thing for an individual to monitor if you take your resting heart rate every morning before you get out of bed, and not including the days that a child jumps up in the bed and screams in your face, but those rare days where you get to just wake up. If you'll take your resting heart rate every day over the course of 6 weeks, 6 months of beginning an exercise program, you will see that your resting heart rate is going down. There will be an increase in intramuscular fat. But you're going to burn that fat. It's not that you're putting on weight. You'll become better at burning fat. And less dependent on carbohydrates. Okay, let me see what I've omitted. Okay, on the anaerobic side, this is the type of exercise that raises the LDL. Remember from over here, the HDL is the one that you want. And let's see, I know I'm still on the small board. Let me give you some numbers here. You want your total triglycerides less than 150 milligrams per deciliter. You want your total cholesterol less than 200 milligrams per deciliter. I think the LDL is 100. Check. Yes. And you want your HDL at least 60 milligrams per deciliter. Now, not every lab, in other words, there's Quest is a big lab in the city. There's another one, the name of which escapes me. Some people go to St. Francis or any part of the Penrose St. Francis system. Some people use the Memorial system. Not every lab is going to have the same exact cutoff, and your doctor may not have the same exact cutoff. For example, to say you want your triglycerides no more than 150, a cardiologist who's treating patients with heart disease very often sees people with this number at 600 or 800. And so that cardiologist is thrilled to death if you get it down to 400. So, some of it is relative, but these are textbook sort of ballpark figures. You will get an increase in muscle mass. This is through hypertrophy. So there's two ways something can get bigger. Cell division, that's not what happens here. In other words, your muscle cells are not mitotically active. That's not why the muscle is getting bigger. Hypertrophy is that the cells increase in size. So the individual cells within whatever muscle we're talking about get bigger, and they get bigger because there's an increase in actin and myosin. There will be an increase in power. Using the word power to mean we could be talking about speed. So if you're trying to get faster because you're going to enter some swim competition, or the increase in power could be referring to strength. There will be an increase in lactate threshold, an increase in the VO2 max, your VO2 max is the maximum volume of oxygen consumed, used by the muscles. So two things about that. Imagine there's a tube in your mouth. You're not measuring how much air goes in. You're measuring how much oxygen goes in. But if you really want to know what was used, you also need to know how much oxygen came out. So if you have oxygen in minus oxygen out, you have oxygen that was actually sent to the mitochondria and used. Yeah, I'm making that point for a couple of reasons. One is we grow up as little kids learning that we inhale oxygen and we exhale CO2. Well, that's true. But you don't exhale pure CO2. There's oxygen in the air that you exhale. If there wasn't oxygen in the air that you exhale, you could not blow on the embers of a fire to get it started. I don't know if you've ever tried to start a fire with wood, camping or in your home, but sometimes you have coals and you blow on them, that gives them oxygen and helps start the fire. Well, if you were exhaling pure CO2, that wouldn't get the fire started. It's further quantified by using its milliliters of oxygen per kilogram per minute. So when we talk about measuring the oxygen in, the oxygen out, we're measuring milliliters of oxygen in 1 minute. Now, making it per kilogram adjusted so that you can compare a 90-pound, 5'9", that wouldn't be a gymnast, what am I trying to say? A 90-pound, 5'1" gymnast to a 250-pound, 6'4" linebacker. I mean, clearly the linebacker has much bigger lungs and is going to consume a lot more oxygen, but by making it per kilogram, you adjust it so it doesn't really matter the size of your body. All right, so that's it for, question? Where am I? What board am I on? Why don't these, Madeline, why didn't you yell at me? And you didn't even chat me, but if you chatted me, I wouldn't know it because there's no little dinger that dings when you chat me. I didn't hear your voice. I'm sorry if you yelled at me. Let me make sure my, hey, say something, say anything.
Anything.
God has spoken himself. Thank you. Your microphone works and so does my speaker. God. Okay, so get that down before I erase it. So allowing time for the people watching the recording to get it. I was thinking about something this morning because in this part of the class, I talk so much about exercise. Guys, if you simply can find the time to walk 20 to 30 minutes a day, 7 days a week, you can't walk 7 days a week, it could be 30 to 40 minutes a day, 5 days a week. Exercising more than 90 or 95% of the population physically fit. You don't have to join a gym. You don't have to buy a fancy treadmill or bicycle. Just take a simple walk. 20 minutes a day, 7 days a week. Shoot for 140, 150 minutes a week. And you don't want to do all of that every Saturday. Okay, every Saturday I'll go walk for 2.5 hours. That'd be better than nothing. But you want to spread it out over, at least probably 4 days a week. Walking with the dog, the friend, the spouse, kids, whatever. Doesn't have to be anything major. Okay, next topic is fatigue. Types and the causes. There are two basic causes of fatigue. First one is the inability to continue sustained contractions. Actually, no. Continue repeated contractions. Inability to continue repeated contractions. I'm going to relate both of these to the running, swimming, biking world as well as the lifting world. This one is easier to understand if we start with the lifting world. So this would be, how many times can one lift the weight? Maybe you're in the gym, maybe you're at home, maybe you've got some simple barbells that you use at home. 1, 2, 3, 4, or 1, 2, 3. Eventually you're gonna, I just can't lift it another time. Done, that's it. As a guideline for anybody that, wants to get started with some of this, you can't, now this would be probably more likely at the gym where you have a lot of control over the amount of weight. But if you can't lift it 10 times, you probably have too much weight set on the machine. If you can lift it more than 12, you don't have enough. Let's say these are biceps curls. Over the course of the weeks and the months, as you get stronger, what you initially could only lift 10 times, you'll be able to lift 12, 13, 14 times, and so you will increase the amount of weight. In the running, swimming, biking world, this would be how long can you maintain an aerobic pace? Missing some words here. How many times lift the weight? Let's add can you or can a person, whatever. How long can someone maintain an aerobic pace? To 80%, maybe. And it might not be that high. If you're walking, it's not going to be that high, but it's still good exercise. Right, the other type of fatigue is the inability to maintain a sustained contraction. Once again, it's easiest to understand what I'm saying if we think about weight. I'm not suggesting that anyone would ever do this at home or in the gym, but how long can you hold the weight? Or there was some old Army movie I watched 10, 15, 20 years ago. For punishment, I had to hold 2 buckets of sand. I don't know what happens if they dropped them. I don't think they got a spanking, but they had to start over and hold the buckets longer. So again, I'm not saying, there's no reason you would do that with weight or at the gym, depending on things you've done in your life, you might relate to it. Personally, what I can relate to is being under a car, holding something up with one hand, and it's right at the limit of what I can hold, and trying to get a nut started on a bolt. And if I don't get it started immediately, I got to let go, rest, and start over because the arm just fatigues. The other thing I can relate to is rock climbing, where you're on a very narrow ledge, basically hanging on with your toenails, and maybe 90% of your weight is on one leg, and you're trying to get a nut and a crack, and you can't, and your leg just starts shaking, and you know you're just done, and you just lean back and say, falling, and hope your belayer is paying attention. All right, so how long can one hold a weight? And in the running, swimming, biking world, this would be just like 1B, except it's an anaerobic pace. Okay. Okay, so now why do we fatigue? What causes fatigue? Well, the first thing that will get you is depleting your stored ATP. That's energy system number one, and you only have 8 to 10 seconds worth of activity using that system alone. But if we hear over the speaker, a missile is about to come through the back wall of E109, you will all die, exit the room immediately and turn left. The speed that you're going to be running at when you jump up out of your chair, you won't be able to maintain very long. Once you deplete that ATP, the stored ATP, you're going to be forced to slow down, and you're going to have to start generating some ATP through one energy system number 2 or number 3. Then if you deplete your stored glycogen, to help make sense out of that, think about aerobic versus anaerobic. The anaerobic systems, you're burning carbohydrates. I told you already, you cannot burn fat anaerobically. Your aerobic system that allows you to burn fat. So if you're operating at a pace, none of this stuff has to be exercise. It can be shoveling snow. You're shoveling snow and there's something else you want to do. You're supposed to meet at 7 and you notice the time. So you start shoveling harder and faster, trying to get the job done. So you've gone into an anaerobic state of metabolism. You deplete your stored glycogen, you're not going to be able to stay in that anaerobic state. You're going to have to slow down. The increase in lactic acid and decrease in intramuscular pH. As we talked about previously, one element there is the pain, which, people talk about embracing the pain, training through the pain, but you can only do so much of that. Eventually, the pH is so far away from the optimal pH that the reactions just won't occur. Your sodium and potassium are out of balance. Under ordinary conditions, when you look at a cell at rest, inside is a high concentration of potassium, and that's relative to the extracellular world where the potassium concentration is low. Inside, there's a low concentration of sodium relative to the outside world where the sodium concentration is high. What we have right there are two sources of potential energy. The potassium wants to move out of the cell, that's down its concentration gradient, and the sodium wants to move into the cell, that's down its concentration gradient. Action potentials are more to it than this, but action potentials are the release of these concentration gradients. So after these concentration gradients have been released, you've got a low concentration of potassium in the cells. It's high outside. The sodium is low outside and high inside. These ions moving back and forth, all of this happening is so fast between the time I touch this, the time I feel it. This happens over 200 times. So it's lightning speed. But once we do this, as we go to the right, we have to go back to the left. Which is recovering. Go to the right, go to the left, go to the right, go to the left, go to the right, go to the left. Over 200 times that happens between the time I touch this and the time I feel it. Last thing, item 19. Isotonic versus isometric contractions. So in isotonic contractions, muscle length is changing. Things are moving. Muscle length is changing. Things are moving. So if you're lifting something, the muscle length is changing. Arms are moving. In isometric contractions, the muscle length is constant. We're not moving. So if I pick this bottle up, that was isotonic. Now as I hold the bottle, it's isometric. Forget the fact that I'm talking, just ignore that. But am I using my muscles right now? Because if I wasn't, I would fall over. So these are isometric contractions that provide me with the stability to stand up. Call it posture. Okay, we are finished with the muscular system. That takes us to the nervous system. So if you look at your copy of the lecture notes, so you can flip back and forth, you see that it's about a page and a half. End this semester with to introduce the nervous system. We don't always get through the entire page and a half. It varies from semester to semester where we stop. But wherever we stop, when this semester ends, it will be just like when we stop today. A little mark so that we know where to pick up on Wednesday. So if we get through everything above that line this semester, then this summer, that's where we will start. If this summer I'm starting here and I get people from another professor's class or another college or university or another state, they're responsible for everything above that line. They have to dig it out of the book, dig it off of the internet. So you'll see these exact 2 pages. Right now, they're the last 2 pages of your lecture outline. But in AMP2, they are a separate document called Introduction to the Nervous System. Essentially, it tells you right there what I just told you. I post on D2L where we will begin here. In other words, maybe we finish with this. So we start with the lecture outline, or maybe we're starting here with Roman numeral 2. I post that on D2L and in one of those early emails that you get, just like the emails you got prior to this semester. I think we may get through the entire page and a half this semester. All right, so first of all, to organize the nervous system, we divide it up into different aspects or parts. And initially, we talk about a central nervous system versus a peripheral nervous system. So the central nervous system consists of the brain and the spinal cord. So the same diagram that I've used repeatedly to represent the axial skeleton. The central nervous system is all about processing information. Like the processors in a computer. I think you all know that a computer has a CPU. Even your watch has a CPU. But a computer has a lot of processing units. It has a visual component, an auditory component. And if you've got a tiny computer, like for example, some of you may have, you've all heard of, a MacBook Air, well, they're so tiny that everything's on the logic board. It's one thing, and if one thing goes, you have to replace the logic board. If you picture bigger computers, like picture a gamer, who's into the fastest games that are out there, they want to be able to replace just their video card, because when a better video card is available, they're going to buy it. Somebody who's using a system to be a component of their video system. They want a separate audio card so they can have the best, most high fidelity, Dolby Digital type stuff. But for everything you can imagine in you, there's a processing unit. And so, for example, I could tell you, I guess I am, that one of the functions of the temporal lobe is hearing. But it's a little more complicated than that makes it sound. There's a part of the temporal lobe, and we'll just call it number 1, that allows you to hear. The raw physiology of hearing. But with that area alone, you wouldn't know what you heard. That's hard to imagine. I'm going to make a noise. You're going to tell me what the noise was. Did you hear that? Yes, Larry, I heard that. What was it? I have no idea. Okay, I'm going to play it again. Did you hear it? Yeah, I heard it. What was it? Sorry, I have no idea. Okay, let me make it multiple choice. Was it a siren, like on an emergency vehicle? Was it maybe a child crying, like they got hurt? Was it a dog barking? I don't know. I have no idea. And that's because it takes another part of the temporal lobe to tell you what you are hearing. And this is just an example to let you appreciate the complexity of things. And there's really not much that we know about it. Because how are we going to find out about it? I know, Raquel, let's go upstairs to the lab and I'll remove a chunk of your brain and see what functionality you lose. That's how we could figure it out. You can't do that. That would, in fact, be illegal. You can do that to dogs. You get limited information. You can do it to primates, get more information than you get from a dog, but you still get limited information. That's expensive. Imagine ordering 1,000 monkeys and you're going to remove a little bit of the, just don't do that. So the way this information is pieced together is from trauma patients, people who've been in accidents, people who've had strokes. So he or she lost this part of their brain due to a gunshot, stroke, stabbed, hit in the head with a crowbar, and they lost this function. You have somebody else, a year later in another state, they were hit in the same place, and they lost the same function. So you say, So we piece it together that way. There's a famous story and famous picture that goes with it that I wish I had. I'd probably find it on the internet. But it's a guy, construction worker. He's working on some scaffolding equipment, and he fell, and a piece of rebar went all the way through his head. He was fine. Well, there was some bleeding that they had to get under control, and they had to remove the piece of rebar, but he didn't lose the ability to hear, see, smell, urinate, any function you want to think of, play the piano. He didn't lose anything. The only effects that were noted, they said he was a little more easily agitated with his wife and children. I guess that's fair. All right, the other thing to say about the central nervous system, and this is regarding the spinal cord, because the spinal cord does not process complex information. The spinal cord by itself processes very, very rudimentary information. For example, I think you all know what the knee-jerk response is if you're at your provider's office and pretend my leg's across, and you strike my patellar tendon ligament, and I extend my leg at the knee. Okay, that's a reflex, and I don't need a brain to do that. Cut my brain off, and you can still, well, I would die. You could still elicit reflexes like that. How do we know that? Because we do it with frogs. We don't actually cut their brain off, we destroy their brain. Bend the head over, and you drive a dissecting needle up through the foramen magnum, and you wiggle it around and just turn the brain into mush. You can still elicit spinal reflexes like that. So very, very rudimentary information, but the cord by itself is processing. But the other important thing that the cord does, and let's get that diagram back up here. It conducts, the cord conducts information to and from the brain. So think about that. If you step on a tack with your right foot, you feel it, don't you? And that perception is in the brain. It doesn't have to be pain. If I touch the board with my hand, I feel it. And that perception of smoothness is in the brain. Well, that signal, that electrical-like signal, had to move not only through my arm, but it had to ascend the spinal cord and go to the brain. Or this signal had to move not only through my leg, but it had to ascend the spinal cord and go to the brain. In fact, if you open my arm and you cut that nerve, then if I touch the board, I would not feel it. Just like if you cut the wire that where you plug the lamp into the wall. If you cut the wire, the lamp won't work because the signal can't be conducted. The cord does process very rudimentary information, but it also conducts information to and from the brain. All right, the peripheral nervous system. Now we're talking about the cranial nerves. I have 12 pairs of those. You know a little something about some of those. In other words, you know the hole they go through. You know that the optic nerve is for vision, not for smell. So they are attached to the brain. The holes in the skull are so that those nerves can get in and out of the cranial vault. And the other nerves that you're familiar with are the spinal nerves. You have 31 pairs of those. And what you should know about them is they pass through the intervertebral foramen, the openings between vertebrae. And I even show you this in the video with 2 vertebrae and a pipe cleaner. They're connected to the cord. I didn't count those. I'm pretty sure there aren't 31 there. So cranial, or rather central versus peripheral. While the central nervous system is concerned with processing information, the peripheral nervous system is concerned with conducting information. Thank God the spinal nerves don't have names the way the cranial nerves do. We refer to the spinal nerves numerically. That would mean the 3rd and 4th cervical spinal nerves. Mean the 3rd, 4th, and 5th cervical spinal nerves. That would mean the 12 thoracic spinal nerves. All right, looking at your outline, you see that the peripheral nervous system is divided into the somatic versus the autonomic. The somatic is voluntary. Some of you are writing or typing right now. That's voluntary. You're moving you're innervating the muscles of the arm to move the hand, to write the letters or strike the keys on the keyboard. This could be walking, snowboarding, throwing a ball, swimming. The autonomic, if you'll misspell and mispronounce the word, call it automatic, it's self-explanatory. It functions automatically. Okay, it innervates all internal organs and functions automatically. You have never in your life I'm going to go outside and get the mail. Oh, the sun's bright. I better constrict my pupils. Come back in. Time to dilate them. Room's on fire. I'm going to run. Increasing heart rate. These things just happen automatically, subconsciously. Now, looking at the lecture outline, you can see that the autonomic nervous system has two sides. The sympathetic and the parasympathetic. It's easiest to understand if you think about the heart rate. Some of you look at your watch. What's your heart rate right now? Mine's 72 beats a minute. What are the two things I can do to my heart rate? Speed it up or slow it down. That's it. So one's sympathetic and one's parasympathetic. Like a light. Turn it on or turn it off. Really, nothing else you can do to the lights in the room. Or you can break them, throw a chair up in the air and destroy them. But aside from that, you can turn them on, turn them off. That's it. You can dilate the pupils, you can constrict the pupils. You can make urine, you cannot make urine. You can make sweat, you cannot make sweat. So almost everything, you've got two sides to it. Now you can memorize charts of what the sympathetic and parasympathetic do. But even next semester, you don't want to do that. I mean, this semester is just an introduction anyway. But even next semester, you don't want to memorize a chart like that. There's no need to. You can use Larry's shortcut. It sounds like exercise. It's a function of the sympathetic division. If it sounds more like something you would associate with rest, it's a function of the parasympathetic division. So when are you going to digest food? While you're doing the incline? While you're sitting down? Sitting down. So most digestive processes are parasympathetic. Increase the heart rate, sympathetic or parasympathetic? Sympathetic. Sweat production, sympathetic. Most of the functions are that easy. They can be derived, and again, that's Larry's shortcut. Here's one for you that might require a little thought. Production of the enzyme lipoprotein lipase. Okay, lipase, ACE, we know it's an enzyme. Lipoprotein lipase. I think I told you earlier this afternoon, that's the enzyme that we use to burn fat. When are you going to be burning more fat? While you're exercising or while you're resting? Exercising. So the sympathetic division promotes the synthesis of the enzyme lipoprotein lipase. The only is an awfully big word, but about the only functions that you can't figure out using Larry's shortcut method are sex. So with sex, you have initial components like the excitement phase to include erection. You have orgasm or climax, and you have resolution. It's all very, very complicated. It does not fit the is it exercise or is it rest. So that's sort of in its own world. All right, cells in the nervous system, Roman numeral 2. So you have neurons. Now you've heard of those. Those are nerve cells. If you go back to a motor unit, we divided a nerve into many nerve cells or neurons. We divided a muscle into many muscle cells or muscle fibers. The motor unit is the single neuron and the small group of muscle fibers it innervates. This is in your notes from the day we talked about characteristics. So it's the same word, neuron. So we know that neurons conduct signals. And we know those signals are called action potentials. Now, sensory neurons conduct signals toward the central nervous system. So I touch this and I feel it, I perceive it as smooth. The signal is having to travel. Well, first of all, I touch this, I feel it, it's smooth. Where do I perceive the texture? My brain. Yeah. Perception is in the brain. So the signal has to be conducted from the tips of my fingers through my arm to my spinal cord and up to my brain. Motor signals leave the central nervous system. I'm going to kick the ball. I'm going to swing the golf club. The signals to innervate the muscles in the arm or in the leg that will affect that particular action originate in the brain. Neurons are funny looking cells. If you've never seen one, you will agree. You may know what they look like from another class. So they have a cell body. Then they have all of those processes. The typical neuron has a lot of short processes called dendrites. And a single long process called an axon. Now, in a neuron, the axon is conducting signals away from the cell body, and the dendrites are conducting signals toward the cell body, just like I've shown already with my red arrows. So signals in these little dendrites are moving toward the cell body, and signals in the axon are moving away from the cell body. Oh, and that little dot or blob in the cell body, what do you suppose that is? The nucleus, the one organelle that you easily see under the microscope in pretty much any cell. We can abbreviate a neuron like this. just a little easier to draw. I'm about to ask you an application question. First of all, remind yourself, which way are signals traveling in dendrites? Toward the body, right? And in axons, their signals are traveling away from the body. So if you see two neurons in series, could be 3 in series, whatever, doesn't matter. Along this pathway, is the signal traveling from left to right or from right to left? Okay, the people saying A are correct. How do you know that it's A? Because the signal in all of these axons got to be going away from the cell bodies. And in the dendrites, the signal is traveling toward the cell body. Okay, so two types of cells, neurons and neuroglia. The neuroglia provide support. Physical and metabolic. Try to do this. God, I've talked before about how the origin of the pain is the head of the femur, but it radiates down through my leg, my thigh, and that's normal. That's what it does. Sometimes all the way to my knee, sometimes to my ankle. Today it's radiating down to my foot. My right foot hurts so bad I cannot wear a shoe. So I have on these cheesy slippers. Don't tell anybody I wore slippers to school. I saw a young man this morning. He had on pajamas. All right, the neuroglia. So the ependymal cells are ciliated cells. Think about the ciliated cells you're familiar with. PSCCE in the trachea, simple ciliated in the fallopian tubes. And in both cases, those cilia are moving something. Trachea, you know, the cilia are moving the mucus layer, and the fallopian tube, the cilia are moving the gametes or the embryo. Well, these ciliated ependymal cells are moving cerebral spinal fluid. Now, you may or may not have heard of cerebral spinal fluid. You probably have, but I'm certainly not suggesting you know anything about it, and you don't need to know anything about it right now. But it is the fluid that the ciliated ependymal cells is moving. This fluid is circulating around your central nervous system, protecting it, cushioning it. What if when you moved your head like this, your brain was bouncing off the inside of your skull? Well, it is, but it's cushioned. So it doesn't feel like, you know, bang. What if I had an egg in a jar? It broke the egg. Well, your brain and your skull, it can't be like that. It's cushioned. So that's one of the things this cerebrospinal fluid is doing. Another way you might relate to it, some of you might have seen a lumbar puncture, some cheesy television show or movie, Grey's Anatomy or whatever, Chicago Med. And the patient is laying on their side with their knees pulled up against their chest, and they stick a needle in around L4. They withdraw cerebrospinal fluid. Plate it out like you do in microbiology and see if there are microorganisms in the fluid. All right, next one, astrocytes. They form the blood-brain barrier. So anything that's going to leave the blood and move into the neural tissue has to be able to cross the blood-brain barrier. This is all about protecting the neural tissue. Of course, you could say, well, it doesn't do a very good job of protecting. It lets alcohol cross, lets heroin cross. The reason those things can cross the blood-brain barrier is because they're so similar to naturally occurring molecules. If we blocked things like, or not we, if the blood-brain barrier prevented things like Percocet, Vicodin, heroin, morphine, Dilaudid from crossing the blood-brain barrier, then the naturally produced substance called beta-endorphin wouldn't cross the blood-brain barrier. Not a perfect system. All right, skip over oligodendrocytes for just a minute. That moves us to microglia. Microglia are phagocytes. So they are phagocytizing debris. Larry, how'd you get debris in your brain? Well, you fell off a cliff and you have an open head injury. You landed in the dirt. How often do you do that? Only twice a day. What other kind of debris might the microglia be phagocytizing? Imagine that you had a stroke. You have dead brain cells. You had a stroke right here in the frontal lobe. Be a high concentration of microglia right here, phagocytizing those dead cells. Skipping over satellite cells, or actually no, not skipping satellite cells. Satellite cells provide physical support. Think of the satellite cells as the styrofoam eggshells that you might use to pack something in a box for shipping. Physical support. Okay, so what that leaves are the oligodendrocytes and the Schwann cells. And I'll cover those sort of at the same time. Look at the notes. You see that the Schwann cells are in the PNS, and the oligodendrocytes are in the CNS. See that right there on the lecture outline. Schwann cells, PNS, oligodendrocytes, CNS. But they do the same thing. So functionally, they are the same. Anatomically, they're different. So what we're looking at here, the picture on the left, this is a neuron, like the one I drew, just prettier. There's the cell body containing the nucleus, bunch of little short dendrites, and a single long axon. Well, what the Schwann cells and oligodendrocytes are doing is they form the myelin sheath. which is like an insulation around axons, sometimes around dendrites, but for the most part, around axons. Insulation. Picture anything in your home that's plugged into the wall, a lamp, a toaster. The wire's insulated. You don't have bare wire plugged into the wall. These are the Schwann cells. They're yellowish. And if we look at this one Schwann cell, we can see that it's wrapped around the axon, forming myelin sheath, which acts as insulation. Now, if we look at the entire axon, we can see that any one Schwann cell doesn't insulate all of the axon. There's a Schwann cell here, there's another one, etc. So it takes many Schwann cells to insulate an axon. You see spaces between the Schwann cells. Those are called nodes. So this is the anatomy of the myelin sheath in the PNS. It's formed from Schwann cells. Once again, looking at the lecture outline, this picture shows us the anatomy in the PNS of the Schwann cells and how they form the myelin sheath. In the central nervous system, we're going to do the same thing functionally. We're going to form insulation, myelin sheath, but the cells doing it are called oligodendrocytes. That word's just fun to say, you must admit. I'd write that word down on the index card that you keep in your junk drawer for neighbors come over, say weird things. Okay, so get your bearings. You can tell these are neurons. You see little short processes, and you see a long process, which is the axon. Here's an oligodendrocyte. So whereas the Schwann cell wrapped itself around the axon, okay, the entire cell is wrapped around the axon. The oligodendrocytes do it a little differently. They send out these processes. So this oligodendrocyte is sending a process over here, myelinating this part. It's sending another process right here, myelinating that part. Here we see the same sort of thing. This oligodendrocyte sending out a couple of different processes. So functionally, we get the same thing. We get myelin. We get insulation. The importance of the insulation will come up Wednesday. But the anatomy, the microscopic anatomy, the way it is accomplished is different. Okay, so your starting point for Wednesday is Roman numeral 3, terms. Madeline, thanks for coming. I'm going to shut it down.